Hello, everyone, and welcome to today's presentation titled Innovating Antibody Therapeutics, How GenScript Enhances Bispecific and TCE Development. This webinar is a part of the 4th annual event in the Immuno-Oncology Virtual Event Series. I am Melanie Chan of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and sponsored by GenScript. For more information about GenScript, go to www.genscript.com. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to first participate by communicating with other attendees using our live chat feature during the presentation. You can find the live chat located at the left of your screen. You can also participate by submitting as many questions as you would like during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We'll answer as many questions as we have time for at the end of the presentation. If you have trouble seeing or hearing the presentation, click on the Help Desk button located at the top of your screen within the navigation bar or from the lobby within the event. Now, let's get started. I now present today's speaker, Amanda Grimm, Senior Segment Marketing Manager of Antibody Drug Discovery at GenScript. For a complete biography on our speaker, please visit the Presenter tab from the menu on the left of your screen. Amanda, you may now begin your presentation.
Thanks, Melanie. I'm happy to be here again. I'm Amanda Grimm. I'm the Senior Segment Marketing Manager for our ADD portfolio here at GenScript. I always enjoy getting to talk to oncology scientists and learn about their projects. So I hope from what I share today that GenScript can help advance your cancer research for improved immuno-oncology therapies. All right, so the contents of my presentation today include a little bit of information about GenScript. We'll discuss a little bit about bispecific antibodies and T-cell engagers. We'll jump into GenScript solutions for helping you with your research, and then I'll close out with a little bit of information about partnering with GenScript. So about GenScript. We have been in development with over 20 years. Of the 20 years of development across various application industries, over 18 of that is in antibody service. We've completed over 150,000 antibody projects, and in addition to that, over 5,000 of them are bispecific projects. The history and milestones of GenScript. We were founded in 2002 in Piscataway, New Jersey, as a gene synthesis company. And since then, we have expanded the molecular biology portfolio. We've added protein expression, the antibody service, cell line development, CRISPR, and much, much more. We've also added additional subsidiaries like BsAb, Legend Biotech, and ProBio. We're really proud of the partnership with Legend and Janssen. They worked on a cell therapy treatment, and that received its first regulatory approvals in 2022. We have a lot of things going on this year in 2025. We've already upgraded and launched new products and products as well. We have a lot more in store, so I encourage you to keep up with us on socials and on the website. This is just an overview of each of those subsidiaries in the GenScript group. I am with the Life Science CRO here in New Jersey. We also have ProBio, our biologics CDMO. Some of the services that I talk about today will be more related to the ProBio side of things. As I mentioned, Legend, developing new cell therapy options, and then we have BsAb doing synthetic biology. All right, let's talk about bispecific antibodies and T-cell engagers. So when we look at cancer in general, the cases continue to rise with many of the different types of cancer. It does remain as the leading cause of death. Some of the early treatment options for various cancers still include chemotherapy, radiation, and surgical procedures. But these early treatments have shown to be less effective. They're not limited to just the cancer cells. And so through our research, we're seeing that antibodies are very promising. The first monoclonal antibody approved for cancer treatment was rituximab in 1997 for treating B-cell malignancies, specifically targeting that CD20 molecule. As investigators continue to study the various cancer pathways and mechanisms, we're able to imagine new treatment routes. These new paths also mean developing new advanced technologies in order to make them possible. So I have a couple of those new antibody therapies that are commonly being explored these days, and that includes the first one here on the left, the antibody drug conjugate. These are really just antibodies that have a cytotoxic drug linked to it, so that when the antibody binds, it's delivering that drug to a very specific target. And then we have bispecific antibodies that are able to recognize two distinct antigens. In this case, one of the antibody fragments binds the cancer cell and the other to the effector T cell, resulting in that T cell activation and localized delivery of the granzymes and perforins that then ultimately kill the cancer cell. So let's look at some of the formats involved in bispecifics. The first one we have, if you look on the left here, are the IgG-like bispecific antibodies. These are similar to natural immunoglobulin. One of the key things with these is that they maintain that FC region. A pro to this type of format is that they have a better half-life and stability due to their complete structure. Also maintaining the FC means that they can engage ADCC, ADCP, and complement activation. When we look at the cons, it's also related to the size, where there's limited tissue distribution, and also there is some complexity in the design. The diagram that I have below just highlights some of the symmetric and asymmetric structures associated with IgG-like. We also have some engineering strategies in that right column. We're not going to really get into those today. We have other resources on our website if you want to learn more about those, but I do just want to call attention to the knobs and hole, the duo body, and the cross map. Those are strategies that GenScript does employ in order to generate its formats. And then over on the right, we have the FC-less bispecific antibody. As the name implies, it lacks the constant FC fragment. That means, as a pro, it's a small size. This allows it to penetrate different tissues. It also has a lower immunogenicity. The con of it, also related to its size, it's less stable. It also has a lower half-life due to this. Again, a diagram just kind of outlining some of the formats involved in the FC-less formats, and a summary of those engineering strategies. Here, if we look at the diagram on the left, we're looking at an image that depicts the mechanism we saw in one of my earlier slides. This really expands on the concept of that one arm fixed to the CD3 on T cells. And then we have the other arm that can be developed to a number of different cancers based on the receptor target. So for example, we can create a breast cancer therapy targeting HER2 in order to bind and activate the T cell. If we look at the figure on the right, these are formats of approved drugs on the market. The top section specifically looking at T-cell engagers. The top one, top left of that is the Rimovab. This was the first bispecific approved in 2009, and it was to target CD3 and EpCAM. Okay, I compiled a list of the approved bispecific therapies for cancer treatments. In this chart, we have about 18. I did pull this information from the Antibody Society. So if you want to go to their website, you can find this information. You can look at much more detail about each of these antibodies that I'm showing today. I really just wanted to be able to highlight the target combinations with CD3 for those T-cell engagers, also highlighting that a lot of these are full-length antibodies, and then we can also see the list of the different cancer indications that they're approved or reviewed for. Okay, let's take a look here. I have an example. We can look a little bit more in depth. That's the Blincyto. This was developed by Amgen and approved in 2014 for acute lymphoblastic leukemia. It targets CD19 on B cells and then the CD3 molecule on T cells. This, as you can see in the upper right figure, it is an FC-less format, so we have just the antigen recognized regions for the anti-CD19 linked to the recognition regions for anti-CD3, and that the small size of this type of antibody drug is going to physically bring the T cells into close proximity with the CD19 positive B cells. So this is a forced interaction that is ultimately going to lead to activation of the T cells independent of MHC or co-stimulatory molecules. This is advantageous since many tumor cells tend to downregulate MHC in order to evade immune detection. We also have a formation of that immune synapse that's then going to trigger the T cells to release the cytotoxic molecules. In general, these kind of processes are highly efficient. One T cell can kill multiple B cells. So when we think about bispecifics, T-cell engagers for antibody drugs, there are some key development considerations. We have the tumor antigen selection, so selecting the right antigen in order to reduce the off-target effects and toxicity. So you want your target antigen to be highly expressed on the tumor, minimally expressed on normal cells. We also have antibody engineering to engineer fragments that can bind the antigens, and this can be done through platforms like hybridoma technology or phage display. We also are looking at the in vitro testing, being able to evaluate the binding affinity, specificity, and cytotoxicity, because we really want to show that that bispecific T-cell engager is going to engage both the T-cell and the cancer cells. For the preclinical trials, we're taking that successful in vitro candidate, testing it in models in order to establish efficacy and safety in vivo. And then you're going to collect that data for pharmacokinetics, biodistribution, and side effects. Right, so with all of that in mind, I'm going to move into talking about GenScript solutions to assist in your antibody drug development. Okay, so here we have a high-level overview of the workflow. It's based on the traditional hybridoma route, just as a basis, but it allows us to divide this into 4 development stages or phases. We have target identification, lead generation and optimization, preclinical studies, and clinical development. And we're going to use this kind of based on those key development areas. We'll walk through this workflow and talk about some of the key products and services that can help you along the way. Here we're expanding those 4 stages and listing out relevant products and services. It's really more for a visual purpose so you can see our coverage. Not everything that we offer is included in this slide, but we will be covering some of these today. Okay, so let's start in the first stage. We have target identification. As I mentioned, with the key considerations, we are thinking about understanding the disease mechanism, selecting that therapeutic target, and then being able to validate your target. And to do that, we have divided this section into 3 application areas where GenScript offers products and services to support your gene analysis, protein analysis, and cell-based analysis. All right, so evaluate functionality and evaluating your target. You can use PCR and or qPCR to amplify your target. We also offer ready-to-use reagents, as well as we have our oligo service where we can create custom probes and primers. They're going to be specific to your gene of interest. Targets can be validated using gene editing to simulate the disease condition or to correct it. So we have a variety of options for off-the-shelf CRISPR-Cas9 plasmids and enzymes in order to support the evaluation of target genes and functionality. Additionally, we can generate functional guide RNA that's going to be specific to your disease and target. Moving into our Gene Titan 8 million oligos, NGS and CRISPR guide RNA libraries. These provide comprehensive exploration of your target genes, giving that precise gene editing and to better understand the gene function. The Gene Titan platform uses semiconductor chip technology to individually build the 8 million oligos in individual strands. These pools contain diverse sequences with user-defined edits that then can be used for your target identification and validation. The next-gen sequencing is something that allows for high-throughput sequencing and analyzing of the genetic material. This information helps to prioritize targets for further investigation. And then we have our CRISPR gRNA libraries. These are cutting-edge technology, and we have a comprehensive collection of guide RNA sequences that target specific genes or pathways of interest. Within the cancer space, some examples are acute myeloid leukemia and bladder cancer. These libraries have been designed and validated by the Broad Institute, and they come ready for lentiviral packaging. Next, we have our CRISPR nucleases. These play a critical role in the gene editing and can help you create a model to better understand the intricacies of disease and the potential of your antibody antigen binding. We offer a variety of off-the-shelf nucleases. As I mentioned, we have Cas9. We also have Cas12a, 13a, MAD7. We have the adenosine or adenine base editor, cytosine base editor, and the prime editor. These can be used to customize your site-specific gene editing strategy with the incorporation of our single guide RNA service. Ultimately, this is going to allow you to create a couple targeted edits, or you can build your own library. It enables high throughput screening of oncogenes or tumor suppressors across multiple cancer models. We can also assist in the right delivery methods, including lentiviral vectors, allowing for the single guide RNA to integrate into your target cell. Using CRISPR has helped uncover molecules in the immuno-oncology field across tumor cells, immune cells, and tumor-immune interactions. Some of those examples include FL1, which is a transcription factor, or TRAF3, a negative regulator for MHC1 expression. For work at your bench, we also offer gels and blotting automation as fundamental tools for protein detection and analysis. This includes the blotting unit, the membrane processing instruments, electrophoresis standards, and high quality antibodies for screening. Automating the Western work saves a lot of time from making the gels, avoiding uneven transfer, and having to handle a million tubes or trays for your staining. We also offer a variety of immune checkpoint proteins and antibodies, which are going to be essential tools for studying the immune system and developing immunotherapies. Taking a look at GenScript's protein expression service, this covers multiple expression systems to accommodate diverse needs and optimize the production of target proteins. Efficient and reliable protein expression is crucial for obtaining sufficient quantities of target proteins for characterization and functional analysis. For simple expression needs, we have the E. coli platform. This provides a cost-effective and rapid solution. It's going to be ideal for expression of soluble proteins, protein fragments, or small-scale production for that initial screening and characterization. We also have an insect expression platform. This is going to offer an alternative for production of challenging proteins that maybe they require proper folding or post-translational modifications. The mammalian expression platform offers you the advantage of post-translational modifications and native folding. So it's going to make it suitable for the production of complex proteins and antibodies that are going to be dependent on proper folding and glycosylation. This type of system really ensures that the generation of biologically active proteins closely resemble their native counterparts. Newly added last year is our cell-free expression platform. This is ideal for those using AI-based drug discovery, enabling you to design, express, and screen faster. It is a great tool in early discovery to test binding and functionality, and is also going to support later lead optimization to precisely control for improved yield, solubility, and folding. Okay, we have our CRISPR cell lines. These are a great approach to genetic manipulation, allowing for the precise and efficient genome editing. These cell line services are going to enable you to generate stable cell lines with precise modifications, such as gene knockouts, gene knock-ins, or point mutations. The cell lines themselves can serve as valuable tools for functional validation, target identification, and even lead optimization. Our scientists use advanced cell engineering techniques to enhance the cell line performance, including increased productivity, improved protein quality, and enhancing stability. In addition to the cell line services, we have a variety of off-the-shelf stable cells. These provide a consistent and reliable source of cells that express protein at known levels. It's going to allow you to evaluate the function and identify potential drug candidates. Within our immune checkpoint cell line collection, this does include PD-1, TIGIT, 4-1BB, and more. Okay, moving into lead generation and optimization, you have now successfully expressed, purified, and confirmed your target. So let's talk about how you can work on generating some leads. This is divided into two sections where we have products and services for lead generation and identification, and then as well for optimization. So to start off lead generation, we can help simplify the gene and plasmid design with our GenSmart intelligent platform, which was actually upgraded to 2.0 last year. This incorporates AI and machine learning into the platform. The codon optimization feature now uses a deep learning powered system in order to enhance the ability to express those difficult proteins, and also is going to result in a significantly higher yield. The construct design tool lets you select from two modes. You can either create a single construct or a library, and in a few steps, you can select the vendor of choice from our extensive library. You then select for the sequence map, where to insert your sequence, and then you would just copy that sequence into the drop-down box. The platform will alert you to any issues that can be addressed, but ultimately you can generate a quote by uploading your information in just a single minute. Integrated into the GenSmart platform is our gene synthesis ordering system. I want to talk about FlashGene. This was launched last year. The great thing about this is that it bundles your gene synthesis, cloning, and plasmid prep so that you receive transfection-ready plasmids for your expression studies. It dramatically accelerates the process to obtain your gene constructs needed for candidate generation and screening, where multiple constructs and rapid cycles of design and testing are required. It was developed with antibody drug researchers in mind, addressing key developmental considerations like promoters, signal peptides, linker peptides, and purification tags. This platform includes gene lengths up to 3 kilobases. We do have over 150 vectors to choose from. For bispecific development in particular, we do have 2 vectors that each address the variable light chain and variable heavy chain. As I mentioned, it's built into the GenSmart ordering platform to create a really seamless process for ordering your genes. We'll take a look now at the plasmid prep service. This is known for its reliability and high quality plasmid DNA. It's really essential for late stage lead generation and optimization. We have options for both research and industrial grade plasmid preps. We can cater to diverse needs in the field, and there's always a guarantee of 100% sequence accuracy. For labs looking for high throughput method for plasmid purification that you can do in-house, we do have Catalog Products Division. They developed their AMAG Quattro. This is a modular designed instrument that's capable of purifying up to 24 samples. Each of those modules can do 6 samples, with all the modules being run and controlled from one controller unit. The modules don't have to be purchased together. These can be purchased individually or together as a package. The automation provides that precise pipetting capability, and you can order the prepackaged reagent consumables. So the consumables are ready to go. You just place them in the instrument along with your samples and select for your program. The runtime is approximately 2 hours, and you can expect yields of 300 micrograms to 1.5 mg of plasmid with low endotoxin levels that are ready for transfection. We have an extensive platform of options in order to design your antibody campaign. We offer the traditional mouse or rodent hybridoma platform with the option to add the monoboost immunization, which would expedite lead generation. Additionally, we have our platforms based on positive cell enrichment and B cell screening and cloning. This includes our rabbit platform, the monorab. This takes advantage of the unique rabbit immune system in order to generate sensitive and specific antibodies with very little cross-reactivity. We also upgraded our hybridoma platform to have the option to offer plasma cell enrichment before the fusion in order to enhance generating specific antibody leads. Both of these platforms have the option to add the beacon B cell screening in order to further expedite the screening process. We do have phage display libraries that support our single domain antibody development using alpacas. This includes access to naive and immunized libraries. And then lastly, if you're looking for fully human antibody discovery, we have the option for transgenic mice as well. As you customize your campaigns, we do support a variety of immunogen types, including protein, peptides, DNA, as well as cell lines. And to expand a little bit further on that slide, I do want to highlight the benefits of mRNA immunization for antibody generation. This particular immunization strategy helps to ensure proper expression of those difficult to express proteins like multipass membrane protein, excuse me. An example of the protein of interest in immuno-oncology is NRP1 with its role in tumor angiogenesis and growth. For labs looking for in-house protein purification, this is our AMAG Quattro 1300 for protein and antibody purification. This is planned to launch here very soon, so this is a little bit of a sneak peek. But we have essentially upgraded our previous instrument to be fully automated with the process and now includes the incubation as well as the purification. It does retain the bead-based purification method. It's high throughput, being able to do about 12 samples in approximately 30 minutes. The 1300 has its own smart pad controller and includes an intuitive interface similar to our Quattro. Our TurboCHO platform is a proprietary protein expression platform developed here by GenScript. It includes the option suitable really at any stage of development with variables based on protein yield, production times, and deliverable characteristics. If we start with the high throughput, this provides transient expression of 1 to 20 mils. It's our fastest at 5 business days, providing you with yields from 0.1 to 10 mgs. This level is most complementary to your early target discovery phases. I also was recently notified that they just upgraded this one to a version 2. So we're actually generating a higher yield and purity with this upgraded version. I didn't get a chance to update the slide before our discussion today, but I did definitely want to share that with you. We also have our express stage of the platform. This is also transient expression, but also at a larger scale of 40 mils to 200 liters. The timeline here is somewhere between 10 to 14 days to provide you with yields from milligrams to grams. It's also capable of providing expression of a diverse antibody format like single domain antibodies and bispecifics across mouse, human, and rabbit. And then we also have our high performance. This moves into the stable expression, scaling up to 200 liters. The timeline here is somewhere between 6 to 7 weeks, and it's going to provide yields ranging from grams to kilograms. The stage supports a later optimization goal in order to generate larger, more stable expression volumes that you would need during preclinical studies and IND. So another option that's not listed here is our fuco-free expression in order to improve antibodies, to provide antibodies with little to no fucose. Fucose can impact the immune response and other pathways. So antibodies expressed fuco-free are able to increase the antibody-dependent cell cytotoxicity outcome by being able to enhance the immune cell like natural killer cells. So if this is important to immuno-oncology research, we can definitely provide those fucose-free antibodies. Expanding on the TurboCHO, we can look specifically at the Express platform. I mentioned it supports a variety of formats that does also include bispecifics. And this is the platform that we use for our bispecific design and development. Our GenScript scientists offer our expertise to support and guide bispecific development. The capabilities incorporate specialized format design, AI-driven codon optimization, optimized production process, optimizing the plasmid ratio, as well as offering multiple purification column options as well. We've completed over 70 different formats, delivered over 5,000 projects, and we've optimized multiple production pathways. Examples of completion I have included on the right. This includes fragments, symmetric and asymmetric as well. You can find more of these on the website, and we do have an interactive feature where you can learn more about the developability and manufacturability for each of them, and that's just highlighted here in the bottom center as an example of what you'll find on the website. We also offer a number of different options if you need to modify an antibody with a conjugate, maybe antibody fragmentation or fluorescent labeling. We can also do single chain variant fragment generation as well. And then if we look at lead generation and optimization, this often involves exploring improving characteristics. So one of the tools that can be used is our mutant libraries. This is a comprehensive collection of pre-designed or custom-designed variant libraries to cover a wide range of mutation types. These libraries provide the ability to screen large numbers of antibody variants, to identify leads with enhanced binding affinity, specificity, stability, or other desired properties. Our precision mutant libraries are powered by our GeneTitan platform that I mentioned earlier. This will enable the improvement of antibody binding affinity via CDR engineering and antibody affinity maturation. This can be done through saturation scanning, site saturation, and/or combinatorial mutagenesis to create a unique library for your project. Next, I want to share our immunoinformatic platform. This is integrated across various services along the discovery and development process. I won't go into too much detail into the entirety of its capability. So if you do want to learn more about this platform, we do have a webinar linked in the booth or on our website. But I do want to call attention to this area, the developability assessment, since we're working through the workflow and we're at the optimization part. This is now part of our TurboCho Smart Quote process. So you would simply just have to select for the report. After uploading your sequence, the algorithm will provide predicted immunogenicity, viscosity, and more. Related to bispecifics, it will also provide a structure annotation of your input sequence. And I do have an example here of what that output would look like. It can accurately identify almost 400 bispecific formats. But in this test case one, we have a sequence where it has recognized the bispecific. It's even identified it as potentially a diabody with a chain ratio of 1 to 1 to 1 to 1. It's also given a structure reference, and that can be compared to this test case 2 on the bottom, where it does not detect the bispecific format, but it does detect a different format of this antibody as an antibody fragment, a single chain, and also gives a structure selection. So now we'll move into the humanization. This technology allows us to optimize antibody candidates for enhanced efficacy and reducing the immunogenicity. Once we have the antibody sequence, this process is based on internal computational algorithms and molecular modeling in order to modify non-human antibodies to make them more compatible with the human immune system while preserving their target binding characteristics. By minimizing potential immunogenicity risks, humanized antibodies have a higher likelihood of progressing through preclinical and clinical development stages. Our service is going to generate up to 15 humanized monoclonals, and our scientists will validate using ELISA binding and EC50 affinity ranking. The top 5 from that will then be provided as small-scale transfection for further validation in your hands. And I did include the icons of the dog and cat so that I don't forget to tell you that we can also caninize and felinize in case you're working in the veterinary field developing therapeutics for our furry friends. All right, so we have antibody candidates. We'll prepare to enter preclinical studies. In this stage, many tests are performed according to FDA, at least in the US, FDA criteria in order to assess and collect data related to toxicity and pharmacogenetics. Non-human animal studies are also incorporated in order to determine whether the antibody performs as intended or whether it supports safe use. So this section is divided into candidate validation and then IND study. I previously shared our plasmid prep production slide, but here I want to highlight our preclinical grade plasmid prep in order to ensure that plasmid production is completed with high standards and extensive QC testing. The chart outlines what you can expect from the service for specific QC parameters and those methods used. Some of the QC examples to note are sequence verification and DNA purification. Anti-idiotype antibodies play a really important role in the development and characterization of your antibody drugs. They are designed to specifically recognize and bind to the unique variable region of another antibody known as the idiotype. These antibodies are generated using advanced recombinant antibody technology in order to ensure high specificity and sensitivity. We also have a large collection of catalog anti-idiotype antibodies as well. These antibodies function to monitor the pharmacokinetics, immunogenicity, and efficacy of the antibody drug candidate in these preclinical models to give the valuable insights into the behavior and potential therapeutic impact. We provide 3 types of these antibodies, those involved in detecting free antibody through antigen blocking. We have the second, detecting total antibody through non-blocking mechanism, and then the last one detects bound antibody, that is drug target complex specific. We also have a comprehensive selection of in vitro bioassays. There are 14 categories and over 40 ready-to-use assays for various targets. Among these include ADCC, that was mentioned earlier as part of the Fuko-free antibody development. We also include immune checkpoint targeted assays, and since bispecifics are the main topic of today's presentation, we can develop specific bioassays for your bispecific T-cell engager project as well. Our gene to lentivirus service is going to generate high quality lentiviral particles that carry your gene of interest, facilitating the functional analysis and in vitro validation of the antibody drug candidates. This service begins with the cloning of the gene of interest into the viral vector, which is optimized for efficient gene delivery. The lentiviral particles are then produced through a state-of-the-art lentiviral packaging system, generating high titer viral stocks. These lentiviral particles can be used to transduce a wide range of cell types, including both dividing and non-dividing cells. So by introducing the gene of interest into target cells, you can assess its functional properties, evaluate biological activity, and study the impact on cellular pathways relevant to the development. On this slide, we have our CDMO ProBio's Biologics CMC service. This offers a comprehensive support avenue for preclinical and clinical stage development. The service is specifically designed to address the critical aspects of biological development, including process development, manufacturing, and quality control in order to ensure that smooth transition from preclinical research to your clinical trials. They work very closely with the clients to develop customized manufacturing strategies that are going to be tailored to the specific needs of that unique antibody drug. The preclinical stage generally requires a large supply of antibodies for in vitro and in vivo studies, including functional characterization, efficacy evaluation, and the safety assessment. The large-scale antibody production service can offer a scalable and customizable approach in order to meet the specific demands of your preclinical research. Our production capabilities can accommodate diverse antibody formats, and the production scientists follow strict quality control measures that offer flexible scalability, competitive prices, and of course, strict confidentiality. So this is the last stage of the workflow and the solutions for clinical development. I've mentioned ProBio already in a few places and want to emphasize that the ease of transition of our technical transfer as the quality scale increases, because we do have our CDMO integrated into our processes. And these final services that I share are provided by our highly experienced CDMO team. The section in particular is divided into cell banking, manufacture, and analytical services. And so we'll go ahead and look at the development support for a well-characterized and scalable cell line that consistently produces the desired antibody with a high yield and quality. This development and cell banking service cover various activities, including the cell line selection, genetic stability assessment, productivity optimization, and establishing of the GMP compliant master and working cell banks. And lastly, our ProBio Pro GMP manufacturing service provides a seamless and compliant pathway for the production of antibody drugs for the commercial use according to regulatory compliancy and cGMP requirements. This covers the whole process of solution preparation, cell culture, downstream chromatography, and filtration using single cell technology in order to avoid any risk of cross-contamination. That brings us to the last section, just to provide you with some information to consider for partnership with GenScript to help you in your research. First, I just want to highlight this achievement. Recently, we achieved over 100,000 citations for our customers' successful research in various peer-reviewed journals. This includes Cell, Nature, and PNAS. And on the bottom here, I just want to highlight the number of citations. They do continue to grow over the years. I'm a little out of date, but we're definitely on track to continue to exceed our goals. If you're curious about what it looks like to work with us, it really just starts with a call to learn about your project and the questions and needs that you may have. The therapeutic antibody development process is complex and likely includes many projects within the scope of what you need. Each partnership is unique and customized to our clients' needs. To do that, we ensure that you have a dedicated project manager to your case to be with you throughout in order to keep you informed, ensure the timelines, and make sure you're getting technical and ongoing support that you need. And with that, I conclude my presentation, and I'm happy to take any questions.
Thank you, Amanda, for your informative presentation. We will now start the Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just submit a question in the Ask a Question box located at the bottom of your screen. Let's get the first question asks, do you work with custom vectors for the Flash gene service?
Yes, absolutely. We do have a vector onboarding process. It does take a few extra days, but once we have that in our system, you're ready to go for any orders after that.
Awesome. Thank you, Amanda. Looks like we have a few questions that came in as well. The next question asks, can the beads be reused?
Yes, this probably refers to the protein purification instrument. Yes, our beads can be reused. They can be regenerated using sodium hydroxide. according to the product specifications. Different beads can be reused different times. So for example, the protein A can be used up to 40 times. So there's a lot of cost savings involved with the beads.
Great, thank you. The next question asks, do you help with the format selection for bispecifics?
Yes, our scientists can help guide you through that process. We have a number of different tools that they use to review your targets and your sequences and can make recommendations for expression as well as purification.
Awesome, thank you. And it looks like we have time for one final question. And that question asks, what AI program are you using for analyzing sequences and developability assessment?
Yeah. So our AI and machine learning tools are internally developed. If you want to learn more about the specifics of how these are used across the immunoinformatics platform, we have a bioinformatics scientist that has provided a webinar that is on demand. It's included in the booth resources as well as our website.
Thank you, Amanda. Do you have any final comments for our audience today?
Yeah, I just want to thank you again for attending. As I mentioned, as I started the presentation, we have a lot of really exciting service launches and upgrades this year. I encourage you to check out the booth. Be sure to check us out on social media where we're sharing a lot of this information. And if you're able to catch us in person at one of our many events, we'd love to talk to you and learn more about what you're doing.
Thank you again, Amanda Grimm, for your time today and your important research. Before we go, I'd like to thank the audience for joining us today and for your interesting questions. I'd also like to thank our sponsor, GenScript, for sponsoring today's webinar. Please visit GenScript's booth in the exhibit hall after the webinar for more information and resources. Questions we did not have time for today and those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand for 1 year until April 30th, 2026. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.